changing that bioanalyte detection into an electrical signal. This electrical signal then is measured using the detector. Does anybody here know what is the most common biosensor used by humans? I'll give you a hint. It's probably used by your parents or your grandparents. Anybody? Nathan, you could say? You don't know? Anybody know? Anybody here have parents who are diabetics? Diabetes in the family? No. Wow. Or one person? Well, Nathan does too. I'm diabetic. And so every night, you know, I poke my finger and I take the sensor and I measure my blood glucose. That's what this is doing. You've got glucose here. There's an enzyme that will react with glucose, producing an electrical signal, which is then given into the detector to give you the glucose concentration within the blood. So that's the most common system used by biosensors. So applications. They can be clinical or non-clinical. Clinical, talking about human diseases. Non-clinical, other applications. Clinical, you got artificial organs, you've got right here glucose monitoring, which we just talked about, but you also have pathology. This is not it. This is a very condensed list. There's a lot more that's possible. People use biosensors also to detect cancer cells inside human blood, to tell them if if a person has cancer. Non-clinical down here, there's a lot of different applications. Again, this is condensed, but you got signal analysis, reactive monitoring, and environmental. My lab, we work a little bit with environmental. We do some reactive monitoring in terms of fermentation processes, but down here we also use bacteria to detect different chemicals that affect the human cell. So we use bacteria in my lab. Why? I'm just going to quickly go through this. The first one, the ubiquitous. What does ubiquitous mean? Anybody? Anybody here? What?
Electric.
No. Good guess. So ubiquitous means everywhere. Bacteria are everywhere. Right now, tabletop, they're there. Your cheek, it's there. Pick your nose, you got bacteria. Okay? They're living organisms, and this is critical because living organisms will only they're the only thing that can tell you, biosensors that can tell you if something is toxic. Other biosensors, glucose monitoring, what does it tell you? How much glucose is there? But it won't tell you if the glucose is toxic. Living organisms are needed to do that. They're easy to work with. They're sensitive, ppm to ppb levels. This is part per million, part per billion. 1 crystal of salt in a liter of water, bacteria can detect it. That's how sensitive it is. Okay? Have short generation times. They grow very fast. They respond quickly. They're genetically malleable. What does malleable mean? Great job. It's easy to mold. It's easy to modify. So bacteria can be genetically changed, recombinant. And there's ethical issues. What are the ethical issues? Do you guys know what ethics are? Basically, good, bad, morality. Out of here, which would you rather die from research? This ugly puppy, this disgusting monkey, this ugly rabbit, oh man, this hamster, this rat, or these very cute bacteria? Which one would you rather have died due to research? The cubic what? Come on, they're cute. No, but that's the point. All of these animals have been used in research. In all of these animals, basically, people have fought for their rights not to use them. Beagles have been used to study gunshot wounds and also chemicals. Monkeys to test vaccines. Rabbits, especially the eye. All the girls here, your makeup, where do they test if the makeup is okay with your eyes? On rabbits. Okay? But all of these tests, or a lot of the tests, maybe not gunshot wounds, but a lot of these tests can also be done using bacteria. And if you kill a monkey, people are like, ah. You kill bacteria, people are like, yeah. Okay? And so there are ethical issues. We use bacteria in our lab so that we basically don't cause any problems. So moving on, how do we use them? Well, we've taken components of other organisms, genetically malleable, and we've put them inside bacteria. One thing that we've done is we've bioluminescence. In all of these organisms here, you can see they're bioluminescent. Not the shrimp here, but the bacteria that it's spitting out of its mouth or inside the water nearby. So all of these are producing light. Biology, luminescence, light. Light produced by a biological organism. Okay? You've got fire, this is not fire, it's not hot. You've got the sun, this is not the sun. This is produced using biological processes. Okay? So shown here is Noctiluca. Noctiluca is a marine organism. I heard you guys studied Noctiluca here. And you can see that as the birds are walking along, it's fluorescent. It's giving off bioluminescence. It's actually not fluorescent, giving off bioluminescence. Likewise, the person here with their hand, rubbing it in water on the beach. And then here, like this one, it looks like he's surfing on electrical currents. You see the bioluminescence coming out. My question for you is, what causes Noctiluca to luminesce? Why is it luminescing in each of these pictures? What's causing it? Huh? I'm sorry?
Physical stimulus.
Excellent. Very good. Here. Physical stimulus. If you look at each of these, what's happening? Physically, the water is being displaced. Why is that important? Because that displacement increases the oxygen concentrations in those localized areas. All right? This here, you like soccer?
Yeah.
All right. Do you know who this person is? Fritz Walker. He's a German footballer. He famous, like my age. He passed away in 2002. But this is football fever. This is called football fever. It's a giant bacteria. Okay? Giant microbe, yes. And so shown here, same idea. This fish here expelling water causes the Noctiluca to basically give off light. And so it uses it as a defense mechanism. It gives off light. The predator trying to attack it is stunned by the light, and it gives this fish a chance to swim away.
Dr. Mitchell?
Yes.
We've seen this actually in other films, not this fish. We've seen the same phenomenon of a marine organism giving off
Light.
light, with bacteria. Is it always Noctiluca, or do
No.
they use different ones? Okay.
No, there's, for the deep sea fish, like you were talking about the anglerfish,
Yes.
inside there and also inside, what's it called, squid, I think that they use, I know the anglerfish, they use, what's it called, Vibrio fischeri.
Okay.
Vibrio fischeri and Vibrio harvii.
And then what about, you'll see copepods actually will fire off a little bomb.
Yes.
Are they using Noctiluca?
I don't know about those, but some of them, I think it depends on which one is nearby. I know for Noctiluca as well, that little squids eat, or little shrimp eat them, but as they're eating them, they're giving off light. And the question for biologists was, why are they giving off light? And it seems to be the response is that as the squid is eating, or the shrimp is eating it, it's giving off light, and it lets a bigger predator know the shrimp is there. So it comes down and eats it, so a little bit of the population is lost, but to the benefit of the whole. All right, so bioluminescence right here. I've already talked about each of these organisms, but one question. What is the one common characteristic in each form of bioluminescence? What is the one common characteristic you see in each of these?
Color.
What color? Blue. All of them are blue. And why is that important? Because blue light can penetrate deeply into the marine ocean. Shown here is the spectrum. And over here is the spectrum that can penetrate in coastal waters. And you'll notice that within the deep ocean, you can get very dark, deep blue color. With coastal areas, you tend to see this muddy brown green. And you can see that in this area here. Anybody know what island this is? South?
South Island.
Of?
New Zealand.
Excellent. Excellent. South Island of New Zealand, right here, Christchurch, and you have this peninsula kind of sticking off. You can see that the water here fits pretty nicely with the color here. Oops. Keeps doing that. Here, anybody know what this is? What island this is? Who said Taiwan? Nathan? Taiwan. This is Taiwan. This is what?
China.
And what is this?
Korea.
And what is this? BFS. All right. And so, but the point here is that near the coast, you see that the water tends to be like this color. But deeper into the ocean, where there's no landmass, it tends to get more blue. And that means that more blue light is penetrating into the water as well. Why is this important? Because what colors do the human eyes see? Red. Does everybody here know Roy G. Biv? All right. Roy G. Biv is basically the, what's it called? The spectrum here. Visible light spectrum. Your eyes have red, green, and blue receptors, which allow you to see all these colors. Your eyes can see more than several million colors, different colors. But deep in the ocean, organisms only have blue-green receptors. They only have blue-green receptors. And so the result is that down in this world, when you watch these BBC programs like Deep Blue World or whatever, I can't remember what the name is, they have pictures of the fish. The fish don't see that. They don't see that. Anybody know what this fish is? All right, Nathan, come on. Right? Stingray. But what are the fish inside? What does a stingray see when it looks at itself? It doesn't see this, because this is like a brownish color, which means it's got red. It's got blue, it's got green, it's got yellow, orange, all mixed in here. But remember, it only, down here, it's only seeing blue-green. So what does it see? Does it see itself? Stingray? This is what it sees. This is actual fluorescence coming off of the stingray. This is blue light hitting the stingray and being transformed into green light. So I'm going to talk a little bit about physics now. Here's physics. Basically light will hit, all right, you can know if it's okay. Light will hit an atom and the electron will be energized. The electron will go up to a higher level. You can have internal conversion where it will come down a little bit to a lower level, but then, oops, I'm not even touching it. But then it will jump down to a lower level. At this point, this green line, it's giving off light. That's called fluorescence. The light is absorbed here, and a different color light is coming out. Now let me ask you, based upon the sizes of these bars, which one do you think is more energetic, the blue or the green? The blue. The blue is more energetic. Which light is more energetic? The blue or violet, green or red? Which one has more energy? How do you know that? What are you more afraid of? Ultraviolet or infrared? Which one causes more damage? Exactly. Ultraviolet. What is infrared? Do you know infrared?
I've only heard.
It's heat. Infrared is heat. Ultraviolet is UV. Okay? And so UV light has more energy, and infrared is less. And so moving this way, you're losing energy in the spectrum. So when you go from blue to green, what happens with the fluorescence? What happens with the light intensity? Not intensity, light energy. It's decreasing. So some energy is lost. Some energy is lost, and that's called fluorescence. So not only do eels and stingrays fluoresce, but also sharks. Swell shark here, you can see the female pattern, male. Very different pattern. The female's pattern tends to be more distinct, more tight. Male tends to be more splotched and spread out. But when you look at them under normal light, it's a lot harder to tell the difference between male and female. They look very similar. But under the fluorescence imaging, what the actual shark sees, it's easier to tell male and female from each other when you look at them from their perspective, not from our perspective. Not only do sharks, but also turtles. Hawksbill turtle underneath the ocean, when you hit it with blue light, you see the shell is now glowing green with fluorescence. So So this is an idea of biofluorescence. So within our lab, and within many labs, what do we want to do? We want to take the genes for bioluminescence and the genes for biofluorescence and put them into our bioreporter, into our biosensor. And the way we do that is we take a stress promoter. This promoter with no stress and with stress, it changes the level of the mRNA. You guys have learned about transcription, right? Yes? What does transcribe mean? Who said that? Excellent. To copy what?
DNA.
No. Transcribe means to write with the same letters. What does translate mean? To write in different letters. So when you go from DNA to RNA, is the language the same? Yes, it is. It's the nucleotide base pairs. The only difference is T to U. But when you go from RNA to protein, is the language the same?
No.
That's why it's called transcription for DNA to RNA and translation from RNA to protein, because you're using different languages. All right? And so showing here, what we do is we take a stress promoter. Do bacteria respond to stress? Yes or no? Are bacteria like, oh, I got homework, oh my gosh. Oops, what happened? I didn't even touch it. Do bacteria respond to stress? Yes, they do. Why? Because they're living organisms. I told you earlier, they're living organisms. And so they respond to stress. Your body, you go out in the sun, UV light, what happens to your body? You burn and then you turn dark. You become tan, right? Bacteria, you put them on in UV, what do? They produce proteins to protect their DNA. So they respond just like we do. So the idea is we take a stress promoter with a reporter gene, bioluminescence or biofluorescence gene, and we combine them together to give transcriptional control of this reporter gene with the stress promoter. The result is that we can see differences in the reporter gene activity when we want it, if there's a stress. So showing here, backtracking a little bit, is GFP. This is the actual protein. It's got a barrel shape. And the part that fluoresces is right here in the gray, and blue and red. And scientists were not happy with just GFP. GFP is green. It's right here, this one. By using mutations of this protein, they've made all of these different colors. They've made all of these different colors. And today I brought some of these. I've got CFP, GFP, YFP. I've got something like deep tomato, cherry, and I've got a really deep raspberry one here for you guys to work with. So what do we do in our lab with bioluminescence? We do the same thing. Stress promoter with the Lux genes that produce bioluminescence. So when the bacteria is stressed, the amount of light coming out increases. Now there's two types of bioreporters. I'm just going to go quickly through this. Basically, these are the stress promoters that you can use. Heat shock, DNA damage, oxidative stress, fatty acid synthesis, so on and so forth. So you can see E. coli can respond to a lot of different stress. Really quickly, when you use an inducible system, the light level will increase and decrease. And I'll show you this in the next picture. So when we add the chemical, the bacteria will start to produce light. This here, you can read this. Can somebody read that? Control. This is a control flask. Do you see any light coming out of that flask?
No.
And this is exposed to the chemical. Actually, if I control the system and I can get it into proper conditions, I can go into a dark room with no windows, or close the door, completely black, shake the flask, and I can read a book underneath it. That's how much light is coming out of our bacteria. And so down here, these flasks down here, less concentration, highest, lower, lower, and even lower concentration with the control here. Okay? And so we can see the response of the bacteria, and that's showing here. Now I just talked about those different genes really quickly, just came through. If you put your hand on the oven, does it turn tan? No? But sun is hot, oven's hot. Why is it, what's the difference? UV. And so your body responds to different stresses differently. Same idea here. TV1061, the bacteria, responds to membrane damage. DPD 2794 responds to DNA damage. DPD 2540, these are the bacterial names, it responds to protein damage. Based upon this right here, what do you think mitomycin C does? Does it hurt membrane, protein, or DNA? Again, membrane, DNA, protein. So which one is being hurt? DNA. The DNA is being hurt. Mitomycin C, anybody know what this is? Mitomycin C is a drug used in chemotherapy for people with cancer. So it attacks the DNA. And so you can see that our bacteria responds to this chemical because the DNA inside the bacteria is being hurt. Likewise, cerulenin. What does cerulenin attack? Protein, DNA, membrane. Right here. Response from membrane damaging agent, cerulenin. And so you see the DNA response, bacteria doesn't respond, the protein bacteria doesn't respond, but the membrane bacteria responds. And so we can tell what stress is being induced. If we take a sample from nature or wastewater treatment plant, we can tell what stress is going to be induced from that. All right, and so this is the opposite one. I'll just skip over that. Don't worry about that. And so to finalize, environmental toxicity monitoring for biosensors, bioluminescent bacteria were initiated back in actually 1995, before 2000, and it's been continuing to this point, and different applications of biosensors in the future. Where it's going to be later, when you guys graduate from college, we have no idea. Right now in our laboratory, we're actually taking our biosensors and we're putting them into human cells to study how the human cells respond to different chemicals and how the chemicals enter the human cells. So it's an exciting area of research. And with that, I'll stop and ask if there's any questions.